So I think we're getting into some pretty serious pharmacology now. Things are heating up a little bit, and we're really starting to get to the meat of the issue when it comes to psychedelics. What are psychedelics doing when they're digging into that 5-HT2A receptor? So in the last video, we discussed two important concepts. One was functional selectivity, i.e. that it's not simply a matter of activating a receptor, but that different agonists can activate the receptor in different ways by inducing different conformations of the receptor, which activates different signaling pathways. And we also talked about receptor crosstalk, and that is when agonists bind to receptors, perhaps two different receptors, it can induce conformational changes in the receptors, which causes those receptors to actually bind to each other, talk to each other, crosstalk, and that this binding interaction can actually have an effect on the signaling pathway. It can affect how one of the receptors, or both of them possibly, affects one or the other's ability to activate its own signaling pathway. And we use the example whereby two receptors binding to each other, one kind of weakened the other's ability to activate its own signaling pathway. But in some cases, you can get enhancement of another signaling pathway. It's a complex and kind of emerging area of modern pharmacology, neuropharmacology, and neuroscience. So you're really at the cutting edge here, people. Okay, so this kind of should give us a little bit of a clue as to what's going on and what perhaps is the difference between LSD, which activates the 5-HT2A receptor and is psychedelic, and Lisuride, which activates the 5-HT2A receptor but is not psychedelic. Now, what people used to think, and some people still do think, actually, I have to be clear that we're into kind of territory where people are still kind of squabbling a little bit, and not everyone's 100% sure. We're going to fall on the side of where I think has the most evidence, but there will be people who perhaps disagree with me, and we simply don't have the time or the energy, people, to get into kind of debates over which is correct. And it is an area, and perhaps in a future offering of the course, we can kind of resolve some of these issues. But originally people thought, well, perhaps functional selectivity provides the answer. So perhaps when LSD binds to the 5-HT2A receptor, it activates the receptor in a certain way and activates a particular signaling pathway, which ultimately leads to the psychedelic effect. Whereas when Lisuride binds to the receptor, even though it's activating the receptor, it's not activating the same signaling pathway as LSD. So maybe let's kind of draw this out to show you what I mean. Okay, so basically all I'm saying is we've got a it's been a while now since I've drawn a membrane, but anyway, so we've got a membrane, and we've got, let's say, here's our 5-HT2A receptor, and when LSD binds to the receptor, it activates a signaling pathway, and we'll call it, for generality, sig path 1, whereas when Lisuride binds to the 5-HT2A receptor, so this is the 5-HT2A receptor, of course, it activates a different signaling pathway. We'll call that sig path 2, and that this maybe is the basis for why perhaps sig path 2 for some reason leads to a psychedelic effect. My handwriting is terrible. Hopefully you know what I'm saying. Psychedelic effect, whereas there's no psychedelic effect when you activate signaling pathway 2. So this was kind of originally thought to be perhaps the key to understanding why lyseride is not psychedelic, whereas LSD is. However, new evidence over the last sort of decade or so suggests actually that receptor crosstalk might be the key here. There might be some influence. It's certainly possible, and there is some evidence that different molecules will activate different signaling pathways, or the balance of signaling pathway activation is different depending on whether it's a psychedelic or a non-psychedelic agonist that binds the 5-HT2A receptor. But it is a bit of a messy issue and remains so. So until that's kind of all tidied up, we will kind of leave that out of the picture a little bit, and we'll focus on what I think there is good, strong evidence for, and that is receptor crosstalk. So what I mean by that is that the 5-HT2A receptor actually interacts with another receptor, and we'll talk about that receptor because that is becoming really, really important in understanding psychedelic drug effects. So before we get to that, I want to just show you some actual experimental evidence that actually shows you the difference in this conformational change that we've discussed between when a psychedelic agonist binds to the 5-HT2A receptor versus when a non-psychedelic agonist binds. So it was always hypothetical up to this point, but then there is actually some decent solid evidence that shows how this works. Okay, so what you're looking at now is perhaps a little bit confusing, so I'll kind of annotate a little bit. This is from a paper published in the Proceedings of the National Academy of Sciences in 2014, so a few years ago now, but very important paper in my opinion. And this shows a part of the, so this is let me try and annotate this a little bit for you. So this is the membrane here. This is the transmembrane domain of the 5-HT2A receptor. You can't see the extracellular domain where LSD is bound. And this here is the intracellular domain. So this is the region, intracellular domain. This is the region here that's going to be interacting with the intracellular signaling pathways. Now, what this study found using combination, I think it was a combination of X-ray crystallography and some kind of modeling, is they found that when a psychedelic drug binds to the receptor, let me think which is the best color here, maybe this green here, you get this particular change. You can see it here in blue. This kind of loop, part of the receptor, kind of twists outward and starts to stick outwards when a psychedelic molecule binds. They describe this here. They've annotated this as HAL, which there means hallucinogenic. We don't really use that term anymore, do we, though, people? But psychedelic, yeah. Whereas here you can see, and I will annotate this in a different color just to confuse you, I'll annotate in red. Here you can see that when a non-HAL, you can't really read that, so I'll write it out, non-HAL, non-hallucinogenic molecules, such as lyseride, binds to the 5-HT2A receptor, you don't get that twisting out of the loop. So when a psychedelic molecule binds, it causes this kind of loop to poke outwards, whereas it doesn't happen with non-psychedelics. And so this indicates the difference between a psychedelic and a non-psychedelic. They're inducing a different conformational change, and that this sticking out domain is actually responsible for the interaction between the 5-HT2A receptor and another receptor, receptor 2. So what is this other receptor that I've been talking about? Well, in an earlier unit, right at the beginning actually, I think in probably unit 1, we were discussing glutamate receptors. Now, we discussed the AMPA receptor as being this glutamate receptor that allows sodium ions to pass through. But there are a number of types of glutamate receptors in the brain. And some of them are not ion channels, but that work in a very similar way to the 5-HT2A receptor and lots of other different types of receptors. These are actually called metabotropic receptors. So these are receptors that we've looked at in detail, receptors where it's not a transmission of molecules through the membrane, but the transmission of information by a conformational change in the receptor. And there is a particular type of glutamate receptor called mGlu-R2, metabotropic glutamate receptor 2, that seems to be crucial in psychedelic effects. And it seems to be the crosstalk between this 5-HT2A receptor and this mGlu-R2 receptor that's important in distinguishing between non-psychedelic 5-HT2A agonists and psychedelic 5-HT2A agonists. So let me explain why on the board. Okay, so here is the M. Why is that in red? I don't want red. I just want a regular. Let's go with sky blue. This is the mGlu-R2 receptor, the metabotropic glutamate receptor 2. So here is the, if I just annotate this a little bit, you can see here is the transmembrane domain, here's the extracellular domain, and here's the intracellular domain. So we would normally draw it, something like this, right? In one of our kind of simplified diagrams. So this receptor, as I said, works in a similar way to the 5-HT2A receptor, but it activates a different kind of signaling pathway. So let's actually have a look at the signaling pathway that mGlu-R2 actually activates and how it does it. So this is a highly simplified, even more simplified than we've looked at before, signaling pathway diagram for the mGlu-R2 receptor. So this is the mGlu-R2 receptor in green. This is glutamate. So now you should be kind of familiar with how these diagrams work. Glutamate binds to the receptor. Now in this case, what happens is normally you have these proteins stuck together when the receptor is inactive. One of them is GI. Now GI. Now remind you, we spoke before about GQ, which was the start of the signaling pathway activated by 5-HT2A. There's another type of really important G protein called GI, which is what we're seeing here. So GI is normally stuck to this other protein called G. It actually stands for G beta gamma. But I've written it just as GBG here. Again, names aren't important. So basically what happens is when the glutamate binds to the glutamate receptor, this particular mGlu-R2 receptor, it induces a conformational change which causes this GI and this GBG to basically fall apart. And so this G beta gamma part of the protein actually leaves the intracellular domain, leaves this GI protein, and actually binds to this potassium channel. Now, where have we seen this before? Of course, we've seen it with the 5-HT2A receptor. However, something different happens. When G beta gamma, this G beta gamma, binds to the potassium channel, it doesn't inactivate it, it actually activates it. It actually causes it to open. It actually activates the channel. Now, what is the effect of that going to be upon the neuron? Well, we should know. This is should be quite obvious to you now, but let's have a look at it. Okay, so let's draw this out again as kind of a bit of revision. So in Unit 5, we discussed the effects of potassium channels and sodium channels on the membrane potential. I'm just drawing out here some of the key landmarks that you will now be very, very familiar with. So what have we got? We've got, of course, we've got the zero voltage membrane potential, we've got the resting potential, and of course we have the all-important threshold potential. This is not to scale. So we discussed basically two types of ion channels, and we talked about this idea that ion channels are kind of fighting with each other to kind of push around the membrane potential, depending on which ions they let through and the direction of in which these ions pass, whether they leave the cell taking charge with them or whether they come into the cell bringing charge with them. So we looked at sodium channels. So sodium channels, what do they do to the membrane potential generally? Well, of course, they depolarize the membrane because they allow sodium into the cell. Because of sodium, of course, has a high concentration outside of the neuron, low concentration inside. So sodium will tend to move inwards through open sodium channels when given the opportunity, and that will, of course, depolarize the cell. Potassium channels, on the other hand, will do the opposite. They will hyperpolarize the cell. Why is that? Well, because potassium has a high concentration inside the cell, low concentration outside. So when potassium channels are open, potassium will tend to move out of the cell, taking positive charge with it, and thus hyperpolarizing the membrane potential. So we've got this fight going on. We've got sodium channels trying to push in this direction, and we've got potassium channels trying to push in this direction. So we saw how when you activate the 5-HT2A receptor, that tends to deactivate potassium channels, which basically allows the sodium channels to kind of win out. So we can actually say here, this is 5-HT2A activation, which tends to push the membrane potential upwards by blocking resulting in the phosphorylation of and inactivation of potassium channels. Now, what we see with the M-glutamate receptor is the complete opposite of this. Doing orange. So with M-glu activation, you see activation of potassium channels, which basically enhances the ability of potassium channels to hyperpolarize the membrane. So potassium channel kind of win out a bit more over the sodium channels, and so we get the pushing in the opposite direction. So this is M-glu-R2 activation. So overall, you can see quite clearly here that the 5-HT2A receptor and the M-glu-R2 receptors are fighting against each other to manipulate the membrane potential. The 5-HT2A receptors are basically trying to push the membrane potential towards threshold by depolarizing, whereas the M-glu-R2 receptors, when activated, are doing the complete opposite. They are attempting to hyperpolarize the membrane, pulling it further away from the threshold potential. Okay, so let's think about this. Let's kind of look at this. Let's look at the kind of the big picture here. So in unit, I forget which unit it was now, but we were talking about the effect of 5-HT, unit 5, of course. So in unit 5, we were talking about the effect of 5-HT2A receptor activation. You activate the 5-HT2A receptor, and that leads to inactivation of these potassium channels, which leads to depolarization. Now, when you activate the M-glu-R2 receptor, in contrast, you're actually activating potassium channels, which leads to hyperpolarization. So we've got this tug of war going on between the 5-HT2A receptor and the M-glu-R receptor, right? So we can kind of see this here. So we can see here exactly what I just said, right? You've got the 5-HT2A receptor, which we've seen inactivates the potassium channels and leads to depolarization of the membrane potential, pushes it upwards, whereas the M-glu-R2 receptor activates the potassium channels and leads to hyperpolarization, or pushes the membrane potential down further away from the threshold potential. So you can see these different types of receptors as kind of fighting against each other, and ultimately where that resting potential, where the membrane potential of the neuron sits, will depend upon the balance of 5-HT2A activation and mGluR2 activation. Now, I've drawn this a little bit differently here to show you something. Now, so this is exactly the same diagram, but I've drawn it slightly differently. I've drawn the 5-HT2A receptor kind of next to the glutamate receptor. Why? Because what seems to happen is that when normally these receptors are kind of independent, they don't speak to each other in any way. So this is where I'm going to introduce the idea of receptor crosstalk. So remember, up in the top right, I just told you that when a psychedelic drug binds to the 5-HT2A receptor, it induces a very specific conformational change. It causes this loop to kind of stick outwards of the receptor. And it's thought, actually, that this causes this change, which is kind of I've illustrated here, actually causes the 5-HT2A receptor and the glutamate receptor to actually kind of stick together and interact with each other. And what actually happens is when LSD and glutamate are both bound to their receptor, they stick together because of this conformational change, and this actually reduces the efficiency of the glutamate receptor in activating its own signaling pathway, which we looked at, and was this GI signaling pathway, which leads to activation over the potassium channels. So what that means is that when LSD, for example, binds to the 5-HT2A receptor, not only does it activate this pathway that leads to potassium channel phosphorylation, this GQ pathway, but it also inhibits the glutamate receptor pathway, this GI pathway. So now we're getting to it, right? LSD, when it binds to the 5-HT2A receptor, actually is able to causes the 5-HT2A receptor to actually inhibit the glutamate receptor. Whereas when glyceride binds to, or serotonin binds to the 5-HT2A receptor, you don't get that loop kind of flipping out, and you don't get that crosstalk between the 5-HT2A receptor and the glutamate receptor. Now it should be coming together in your head. However, I think I'm going to give you a little bit of time to kind of digest that. So please watch this again if you've missed the point here. Hopefully I made it reasonably clear, but you might want to watch it again. And in the next video, which might be the last video of the unit, it might not be, I'm going to go through this again and think about the consequences of this receptor crosstalk between the 5-HT2A receptor and the glutamate receptor, and why that can completely explain why glyceride is not psychedelic, but LSD, psilocin, DMT, etc. are psychedelic. So, take a deep breath. It's getting hot here in Okinawa. It's getting hot here in Okinawa. It's getting hot here in my flat. So I will see you in the next video. Bye bye.